Today we're looking at a new Nature Communications paper on coffee. This is actually a proper human intervention study, and it shows that coffee doesn't just act as a stimulant, it reshapes the microbiome and links to behaviour through the gut-brain axis. We'll go through the data, where it sits in the field, and what this tells us about how experimental design maps onto journal tier. I'm Dr. Susan Heavey, Associate Professor of Translational Medicine at UCL. Welcome to Further Reading. As always, I'll give you a brief walkthrough of the paper, followed by a figure-by-figure discussion, and then some bigger picture paper craft items. So firstly, a summary. This study shows that habitual coffee consumption reshapes the gut microbiome, alters metabolite profiles, and is linked to measurable changes in behaviour and cognition. Coffee drinkers had distinct microbial species and reduced levels of certain neuroactive metabolites, such as GABA, alongside higher impulsivity and emotional reactivity. Importantly, many effects changed with withdrawal and reintroduction, and some occurred independently of caffeine, suggesting coffee influences the microbiota gut-brain axis as a complex mixture, not just a stimulant. Jumping into a relatively concise figure-by-figure overview, Figure 1, as it often would, gives you an overview of the study design and shows some behavioural outcomes. This shows us that it's a controlled human study, comparing coffee drinkers versus non-drinkers, followed by withdrawal and reintroduction. That's for both caffeinated versus decaf. The key finding is that coffee drinkers show higher impulsivity and emotional reactivity at baseline. Both drop with withdrawal and improve further after reintroduction. Caffeinated coffee improves anxiety and some cognition, while decaf preferentially improves memory and sleep-related outcomes. Figure 2 is around fecal metabolomics. We see here that coffee consumption shifts gut metabolite profiles. The key shifts are some increases in caffeine-related compounds like theophylline, hypuric acid, and decreases in neuroactive metabolites like GABA and indole derivatives. Now, these changes partly reverse during abstinence and respond differently to caffeinated versus decaf reintroduction, showing both caffeine-dependent and independent effects. Figure 3 shows us another heat map, and this one is around gut microbiome composition. Here, we see that coffee alters specific microbial species rather than overall diversity. The key finding is that there is an enrichment of species like Cryptobacterium and Egerthella in coffee drinkers, with shifts reversing during withdrawal and reappearing after reintroduction, even with decaf. This indicates targeted microbial changes driven by coffee components beyond just caffeine. Now in Figure 4, it's a trendy Sankey plot. Here we see the integrated microbiome metabolite behaviour network. This links microbial species to metabolites to cognitive or behavioural outcomes. The key finding is that specific metabolites, such as theophylline, caffeine, and phenolic acids, act as hubs, connecting microbiome changes to cognition and behaviour, for example, stress and memory scores. Now, this supports a mechanistic microbiota gut-brain axis influenced by coffee. So this is all supportive evidence building up that picture around the gut-brain microbiota axis. It's not causative, but there's some interesting correlations and links going on here. Figure 5 establishes that coffee consumption affects fecal polyphenolic metabolite secretion. We see that coffee drives changes in these phenolic metabolites derived from diet. This key finding is that coffee increases specific polyphenol-derived metabolites. These drop during abstinence and rise again after reintroduction, regardless of caffeine content. This reinforces that non-caffeine compounds, polyphenols, are major drivers of gut effects within this study population. Figure 6 goes on to connect the shared impact of coffee on microbial species and urinary polyphenol metabolites to influence cognition and behaviour. Here they're tracking systemic metabolism of coffee compounds. The key finding is that caffeine-derived metabolites return only with caffeinated coffee, whereas phenolic metabolites increase with both caffeinated and decaf. These metabolic signatures correlate with microbial species and cognitive outcomes, highlighting strong inter-individual variability driven by the microbiome. To understand where this paper sits, you need to think about how the field has evolved over the last few years. Firstly, there's been a major shift from correlation to mechanism in microbiome research. Early large-scale studies like the Zoe dataset showed that diet, and particularly coffee, is strongly associated with gut microbiome composition across tens of thousands of people. But those studies were observational, so they could tell you what correlates, but they couldn't tell you what causes. Remember, correlation does not equal causation. Second, the field has converged on the idea of a diet microbiota gut-brain axis. Recent high-impact reviews in Nature Metabolism and related journals emphasize that dietary components reshape the microbiome, which in turn produces metabolites that influence brain function and behavior. Mechanistically, this operates through immune, neural, and metabolic signaling pathways, things like short-chain fatty acids and neurotransmitter precursors. Third, there's growing interest in polyphenols as microbiome active compounds. Reviews highlight that plant-derived molecules like those in coffee are metabolized by gut microbes into bioactive compounds that can influence cognition and inflammation. So with all of that in mind, where does this paper fit in? Well, it essentially bridges each of these three layers. It takes a known dietary exposure, so coffee, and then moves beyond association into a controlled human intervention. Then integrates microbiome, metabolomics, and cognitive outcomes in the same individuals. It's a super nice design, and that's the key advance. It's not proving a new concept, but it is connecting diet to microbiome, to metabolites, to behavior in a single experimental system. So, in the wider literature, this paper is best seen as a mechanistic, mid-scale human study that operationalizes the gut-brain axis in practice. It doesn't discover it, but it's a really nice next step. I think having situated where these results sit within the wider literature, it's a good time to focus in on the experimental design that was employed here. I think that the design that they used was genuinely elegant, actually. They're trying hard to disentangle one of the really tricky problems that's there in human nutrition studies at the moment, and really across a lot of studies at the moment, and that's causality versus correlation. Here they start with a clean cross-sectional comparison, habitual coffee drinkers versus non-drinkers, matched on diet and baseline characteristics as best they can. But they don't stop there. They embed this within a longitudinal intervention in the same individuals. So first a controlled 2-week withdrawal, then a randomized, double-blind reintroduction of either caffeinated or decaffeinated coffee. That combination is a key strength. I really do think it's very nice. It gives you 3 layers of inference. You've got between-person differences, within-person changes, and causal separation of caffeine versus non-caffeine components. It's all pretty nicely controlled throughout. Then they pair this design with deep phenotyping across systems. They've got shotgun metagenomics, which is exactly what you would hope to see, targeted and untargeted metabolomics, which is also key. Then ensuring that it's just not purely focused on the biology and the omics at that microbial and molecular biology scale, but also looking right the way up to cognitive testing, behavioral questionnaires, and also immune profiling. All of these things sampled repeatedly across the timeline. It's complicated, but needs to be. It's expensive, but needs to be. And it really does produce quite a nice paper in the end. It means that you're not just observing associations, you're watching the microbiome, the metabolites, and behavior shift dynamically as coffee is removed and then reintroduced. Finally, they integrate those layers using multi-omics modeling to link microbial species, metabolites, and cognitive outcomes directly. The result is a study that doesn't just ask whether coffee matters, it shows how, through the gut microbiota-brain axis, and crucially, which effects are caffeine-dependent and which are not, which of course is a question on most readers' lips as they're reading it. Now, one thing I get asked a lot is how to know what journal to pitch a paper to when you're writing it. And I think you can learn that from reading as well as from anywhere. Here's an example where I've laid out that they have a really nice, elegant experimental design. They've spent a lot of money on some really cool analyses. They've got a good sized cohort. Everything's well designed, well controlled. So why isn't it in Nature Cell Science? Why is it not in a top tier journal? Now, that's definitely not to throw any shade at Nature Communications, of course. This is still a really good journal and a really good paper, but why is it not quite getting to that top level? Okay, I think in the bigger picture, let's use this as an example. It could be a way that we can think about journal tier while we're reading future papers. So let's start with the design logic. Is the study just observational, or does it include within subject intervention, randomization, blinding, causal inference? That's your first signal. Then start to look at things like scale and robustness. Are we talking a handful of patients, tens, thousands, multiple cohorts, multiple nationalities? High-impact journals tend to want either large-scale validation or very strong, clean effects. The third thing to look at is mechanism versus association. So does the paper just show correlations, or does it actually demonstrate a causal pathway, ideally with targeted manipulation? And finally, conceptual advance. Ask yourself, does this fundamentally change how we think about a biological system? Or is it refining something we already broadly understand? Is it taking a minor incremental step forward in a field, adding another brick in the wall of evidence, or a giant leap forward? So, back to this specific paper. In a paper like this, you've got beautiful design, multi-omics, pricey, causal elements. It sits very comfortably in this nice level of Nature Communications. To move up to, say, Nature itself, you'd usually either need bigger scale, stronger causality, which is beginning to be established here, but not really beyond all doubt, or perhaps a clearer step change in biological understanding. Remember, each of the sort of links in this paper along that microbiota gut-brain axis are things that there's already a growing body of evidence to support. This paper doesn't change everything, but it reinforces that the field as a whole is on the right track with regard to it. So good experimental design does get you into a top journal, but decisive mechanism or conceptual novelty is what would push you into the very top tier. Before we go, let's do a little data Viz Corner just to help you with reading a specific type of plot. Here, I'm going to use figure 2 as an example of how to read a Metabolomics heat map if you've not read one before, or if you have but you're worried you might have been spoofing. You probably weren't, but let's double check. Start with the structure. Each row here is a metabolite, things like caffeine, theophylline, or GABA. Each column is a condition. Non-coffee drinkers, coffee drinkers, then the same individuals after withdrawal and after reintroduction. Now, look at the colour scale first, not the numbers. Red means higher relative abundance, blue means lower, and white means no change. And importantly, these are effect sizes relative to baseline coffee drinkers, not absolute concentrations. Then move row by row, not column by column. Pick one metabolite, for example GABA, and track it across conditions. You can see it's lower in coffee drinkers, then shifts again with withdrawal and reintroduction. That tells you the direction and reversibility of the effect. Next, check for consistency across participants. Those small horizontal bars within each cell show individual data points. If they're aligned, the effect is consistent. If they're scattered, there's variability. Finally, ask the key biological question. Do changes reverse with withdrawal, and do they then return with reintroduction? That pattern is what begins to give you causal confidence, not just the presence of red or blue. So the principle here is, don't just read colour, read trajectory, consistency, and reversibility. If you found this useful beyond trying to decide whether or not to have that next cup of coffee today, well, I would say try applying this as a sort of framework to the next paper that you read. Look at the design first, then the data, then the claims, because that's what can really separate surface level reading from actually understanding the literature. If you'd like to look at other paper summaries to keep up to date with the literature, followed by these little peppered through bits of paper craft and broader thinking in academia, then you can find lots of videos like this across our channel. So please do have a look. And if you've made it this far into the video, well done and happy reading.